I'm Eva. I'm in section 16. I am wanting to be a PA, so that's why I'm taking this class, and I'm really enjoying the class so far, except for the midterm. And yeah.
Hey, I'm Jose. I'm in section 9 of the lab. I'm trying to go to med school, so that's why I'm taking this class. And I think it's actually been pretty cool. I learned a lot of procedures I wouldn't have learned if I hadn't taken the class. So yeah.
My name is Josh. I'm in section 11. I'm a bio major, and I want to go to medical school after I graduate. I'm not really enjoying class, but yeah, I hope I can do better.
Hi, my name is Justin. I am also a bio pre-med major. I'm in section 11 as well. My TA name is Nick. He's alright. He's a pretty chill guy. What else? My career. I would like to be a neurosurgeon after college, and that is what I'm starting to do. In class so far, I'm enjoying it. It's okay. Midterm.
My name is Serenity. I'm in section 11. I'm in animal science, science and technology concentration. I hope to be a veterinarian when I finish college, and I am enjoying the class.
I'm Zayla. I'm in section 11. My major is kinesiology to be a PA, and I'm loving the class. You're helping me so much. Okay.
And we are virology.
Yes.
Oh, we're also doing chapter 9?
Yes.
Chapter 9.
Alright, should I start?
Yeah.
Okay. So, chapter 9 includes mutations and mutants, molecular basis of mutation, reversions and mutation rates, and mutagenesis. So, bacteria can exchange genes, and mutation is basically the heritable change in genomes, and it can lead to a change in properties of an organism, and some beneficial and some detrimental, but most have no effect. Exponential growth in prokaryotes accumulates mutations quickly, and horizontal gene transfer, or genetic exchange, generates much larger changes. And then mutation and genetic exchange fuel evolution. So genomes of cells are double-stranded DNA, and viral genomes are double or single-stranded DNA or RNA. There's also wild-type strains, which are isolated from nature, and can also refer to just one gene. For mutants, a cell or virus derived from wild types that can carry a nucleotide sequence change. And then genotypes designated by 3 lowercase letters followed by a capital, and they're all italicized. Observable properties may also be altered. So it's a phenotype, and they're designated by capital letters and 2 lowercase letters, and then a plus or negative. Examples of phenotypical traits include glucose fermentation, production of enzymes, etc., and can be attained either from wild type or parental strains. So isolation of mutants is screening versus selection, and selectable mutations confer an advantage under certain environmental conditions. Mutated progeny cells outgrow and replace parent. For example, antibiotic resistance. They're relatively easy to detect and powerful genetic tools used in genetic engineering. And for screening, its non-selectable mutations do not confer an advantage, even though they may lead to a phenotypic change. And for example, color loss in a pigmented organism, and requires laborious, time-consuming screening. And also, its blue or white colony system in bioengineering requires screening, which is time-consuming.
So here's some differences between screening and selection. So you're gonna use understanding physiology is key to designating the genetic screens, and you're gonna use selection over screening whenever it's possible. I'm gonna give you some examples of common types of mutants and their detections. So an auxotroph, it has the inability to grow on a media lacking select nutrients like amino acids. If it's pigmentless, that means that a loss of an enzyme in a biosynthetic pathway causes a lack of color or a different color. Sugar fermentation occurs when there's a degradation pathway. It doesn't have any color change and it has a solid medium. There's also going to be spontaneous and induced mutations. Spontaneous occur without intervention, so like the DNA doesn't correct it. Most of them are just like errors in the DNA polymerase during replication. And then induced mutations are usually caused deliberately, like in a lab or in a controlled environment by a researcher. And that's like exposure to natural radiation or chemicals that modify DNA. And then there's a point mutation, which just changes one base pair, or a base pair can change stay the same, or two base pairs could change. But yeah, and it's only single base substitution, and it can cause a phenotype change or a genotype change. Then there's some other forms of base pair substitution, like missense, nonsense, and silent mutations. Missense is basically, it just changes one of the sequence of amino acids, and it doesn't really it doesn't really lead to like dysfunction or anything, but it can. And those are like transitions with purines and pyrimidians, and transversions with pyrimidians and purines. Silent mutations don't really affect anything, it just changes the letter. And nonsense mutations, they usually make it to where it's not functional because it's too truncated or incomplete. And then there's frame shift mutations, and that's going to be whenever a deletion or an insertion occurs, but it changes how the sequence is read. So if it was like cat saw the rat, you would delete the S essentially, and that's going to cause lethality, or it could be like a loss of the gene's function.
I think I'm 14.
Okay, so mutation rates depend on frequency of DNA changes and efficiency of DNA repair. Reversion occurs because point mutations are typically reversible, and a revertant is a strain in which the original phenotype is restored. Mutation rates for most microorganisms occur in DNA replication at a frequency of 10 to the negative 6 to 10 to the negative 7 per kilobase. Eukaryotes have a 10-fold lower error rate than microbes. DNA viruses have error rates 100 to 1,000 times greater. RNA viruses error rates even higher due to less proofreading and lack of RNA repair mechanisms. An induced mutation is an environmental factor that causes mutations and can be imposed by the researcher. A mutagen is a chemical, physical, or biological agent that induces a mutation. Some examples of chemical mutagens are nucleotide base analogs, and this is when it resembles the nucleotide bases but have faulty base pairing. An example of this is when aminopurine is substituted for adenine. Another example of a chemical mutagen is, chemicals that cause modifications to A, T, G, and C. An example of this is an alkylating agent, which introduces changes in either replicating or non-replicating DNA, and it results in incorrect base pairing or single base substitution. Chemical mutagens that cause frame shift mutations as well. And an example of this are intercalating agents, and they insert themselves between two bases, pushing them apart, triggering insertions or deletions. So have physical mutagens, and one of them is a non-ionizing radiation. An example of this is ultraviolet rays, I guess. They have lower frequency. Purines and pyrimidines strongly absorb UV, and they primarily form pyrimidine dimers. That's a kink in the DNA structure. Ionizing radiation have higher frequency. They're more powerful than UV. They ionize water and other substances, and they result in free radicals such as hydroxyl radical that damages macromolecules, and they cause double or single-stranded breaks and can lead to rearrangements or large deletions. So the DNA repair in the SOS system, so mutations are heritable, and if damaged, they can be corrected before cell division so that no mutation occurs. And in bacteria, stalled replication or major DNA damage activates the SOS repair system. It initiates many DNA repair processes, some of which are error-free. It allows DNA repair to occur without a template. For example, with a random incorporation of DNTPs, and it results in many errors and mutations. And in E. coli, SOS controls transcription of about 40 genes. Regulators are LEX-A and REC-A proteins.
Genetic recombination is a horizontal gene transfer, which is gene movement between cells that are not direct descendants. They allow quick acquisitions of new characteristics, fuels metabolic diversity. 3 mechanisms of bacterial genetic exchange is transformation, transduction, and conjugation. 3 fates: degradation, replication by itself, recombination with host genome. Recombination is physical exchange of DNA between genetic elements. Homologous recombination is a process that results in genetic exchange between homologous DNA from 2 different sources. It drives crossing overs in classical genetics. Rec-A helps recombination to occur. Effects of homologous recombination on genotype: the sequences must be related but distinct. In bacteria, genetic recombination occurs after fragments transferred by transformation, transduction, or conjugation. To detect exchange of DNA, recombinant cells must phenotypically differ from both parents. In lab experiments, usually use recipient strains lacking a selectable characteristic that recombinant gain. Complementation occurs if a functional wild-type copy is supplied on a plasmid or bacteriophage, restoring wild-type phenotype. Transformation, genetic transfer process by which free DNA is incorporated into a recipient cell and brings about genetic change. For example, cell lysis. Typical transformable size is 10 genes or 10,000 nucleotides. Only a small proportion of genes transferred in one event. Competence in transformation. Competent, an acceptor cell that can take up DNA and be transformed. In some bacteria, competence directly linked to pili or secretion system proteins includes 3 stages: binding of DS foreign DNA, uptake of DNA, Rec-A mediated homologous recombination of a new strand into bacterial genome. If the foreign DNA is homologous to host DNA, recipient cell is considered to be transformed. Plasmid DNA must remain DS and circulate for replication. Transduction, transfer of DNA from one cell to another bacteriophage. 2 modes. Generalized transduction, DNA from any portion of the whole genome is packaged inside the virion. Donor genes cannot replicate independently, will be lost without recombination. Sometimes host DNA accidentally package its phage, forming transducing particles that is defective and cannot lead to virolytic infection. Specialized transduction, DNA from a specific region of the host chromosome is integrated directly into the virus genome, typically replacing some viral genes. Homologous recombination may occur or may be integrated during lysogeny. which is a rare event, part of host DNA incorporated into fixed DNA, all favors a very high efficiency of transducing a cell. It occurs in many bacteria and at least one archaea. For example, multi-antibiotic resistant genes in Salmonella, Shiga-like toxins in E. coli, virulence factors in Vibrio cholerae.
Conjugation is horizontal gene transfer that requires cell-to-cell contact. There's an F positive cell and an F negative cell. The F positive cell is the donor cell, and the F negative cell is the recipient cell. Only the donor cell contains the conjugated plasmid or the F plasmid. Other genetic elements are also mobilized. It takes about 5 minutes for an acceptor cell to become F positive, and an F apostrophe, a fertility plasmid. It contains the genes that regulate DNA replication and contains transposable elements that allow the plasmid to integrate into the host chromosome. Transfer of chromosomal genes to the F plasmid. Sometimes F plasmids can be excised from the chromosome, sometimes incorporating chromosomal genes, which makes the F prime plasmid. F prime plasmids are F plasmids that contain those chromosomal genes. They transfer chromosomal genes to recipients at a higher frequency, and F prime mediated transfer resembles specialized transduction. Horizontal gene transfer in archaea. Most archaea contain a single chromosome like bacteria do, and genetic manipulation of archaea is less developed than in bacteria. Most archaea are extremophiles that grow under high salt or high temperature, and most antibiotics do not affect archaea, so choice of selectable markers is really limited. There's no single species model for archaea like there is for bacteria, but there are various transfer mechanisms like transformation, transduction, and conjugation, and several plasmids have been found. For example, transformation occurs in Methanococcus fultae, Pyrococcus furiosus, which is they have an interspecies plasmid change in Thermococcus and Halorubrum through cell envelope budding, but transduction is rare. Conjugation in archaea is the same as bacteria, it's cell-to-cell contact, but pillae are not formed and it's still unidirectional DNA transfer. Mostly sulfolobus can exchange DNA by cell aggregation occurring when pillae synthesis is triggered by UV radiation.
Mobile DNA is segments of DNA that move as a unit to another DNA. Most mobile DNA are transposable elements, meaning they have no origin of replication. They move by transposition, recognition, cutting out, and ligation into new. They're abundant and found in all three domains of life, viruses and plasmids. Two main types of transposable elements in bacteria are insertion sequences and transposons. They encode transposers. Enzyme required for transposition. They have short inverted repeats at ends, also required for transposition. When inserted, a short sequence in target DNA at integration site is duplicated. Cells have mechanisms or immune systems to prevent horizontal gene transfer and viral. Non-specific is analogous to innate immunity, and specific is adaptive immunity. Bacterial and archaeal innate immunity is restriction endonucleases protect cell from viral attack and transformed or conjugated DNA. Chemical modification protects cells' own DNA and sometimes allow viruses to avoid restriction endonucleases. In the bacterial and archaeal innate immunity, that's phage exclusion and abortive infection. Phage exclusion is a variant of restriction enzyme systems that recognize and modify incoming foreign DNA and they prevent replication. Abortive infection triggers host suicide, which leads the cell to death by the toxin, antitoxin systems, and they prevent more virus from being spread. The adaptive immunity, and CRISPR is the clustered regulatory interspace short palindromic repeats, which is a major antiviral defense system in both bacteria and archaea. It seeks and destroys foreign nucleic acid. It's a short repeat of constant DNA host chromosome sequence that altered with short variable DNA sequences or spacers that previously invaded the cell. Spacers are memory bank and incoming nucleic acid for memory cells. The specificity makes the CRISPR analogous to adaptive immunity. For them to I was doing one slide.
No, you're good.
For it to work, the spacer must already be present. Immunization probably occurs upon contact with inactivated virus or if restriction enzymes cleave invading DNA. They're widely distributed. Viruses have evolved mechanisms to avoid it. And that's it. The rest is just pictures.
Perfect.
Is that enough time?
Yeah, and then we have to show the.
Oh yeah, that's okay.
This is our concept.
All right. Thank you.